Hello, this is Professor White, and today I'll be talking about the cell membrane and transport. So this is just a basic understanding of what cell transport is for this lesson. Now, first and foremost, let's look at the function of the cell membrane. As you know, the cell membrane separates the components of the cell from its environment and surrounds the cell. It is kind of like a gatekeeper of the cell, which regulates the flow of materials into and out of the cell, which is why it's called selectively permeable. Now, the cell membrane helps cells maintain homeostasis, which is kind of like a stable internal balance. Now, looking at what homeostasis is, the cell membrane is responsible for maintaining the balance within the cell, and therefore when you say homeostasis, it is a stable internal environment. And the cell membrane maintains the homeostasis through balancing the pH, the temperature, glucose, sugar intake, and also water balance. Now, it does this through active and passive transport. Now, going back to the parts of the cell membrane, also known as your phospholipid bilayer, all cells have cell membrane made of phosphate, proteins, and lipids. That's why it's called phospholipid bilayer. So, as you can see here in this diagram, you can see that there's like a protein channel stuck in between those bilayer, and there's also this protein pump. Now, all cells have plasma membrane. Prokaryotes, on the other hand, have a cell wall and cell membrane. Now, eukaryotes, for animal cells, they only have cell membrane, and for your plant cells, they have cell membrane and a cell wall. Now, let's take a look at the cell membrane in detail. Now, it's a double layer, that's why it's called bilayer, of phosphates and fats. Now, a single phospholipid has a hydrophilic or water-loving phosphate head and hydrophobic or water-hating fatty acid tails. Now, the cell membrane both repel and attracts water through the membrane at the same time. Now, let's take a look at the different cell transport, and we'll start first with the passive transport. Now, passive transport is a process that does not require energy to move the molecules from high to low concentration, and we have three: diffusion, facilitated diffusion, which uses a protein to push particles across, and osmosis. So let's take a look at first diffusion. Now, diffusion is a movement of small particles across the cell membrane, like the cell membrane, until we reach homeostasis. Now, facilitated diffusion requires the help of a carrier. Usually they are channel proteins. Now, these particles move from an area of high concentration to low concentration, which is why it's called passive transport. Now, some examples of diffusion, you have those aerosol spray and perfume, wherein inside the can, you have a high concentration and the molecules are packly tight together. As soon as you spray the aerosol, now it goes into a low concentration outside the can, which cause these molecules to spread out. So again, diffusion, high to low concentration. Now let's go to osmosis. Now, the osmosis, on the other hand, is the movement of water through a selectively permeable membrane like the cell membrane. Now, the water moves across the cell membrane from an area of high to low concentration as well. But as you can see in here, in this particular diagram, you see that red broken lines. That is your semi-permeable membrane, wherein on the left-hand side, you would see the water actually going in to the other side, the right side, because there's low concentration of water. Semi-permeable membrane is permeable to water, but not sugar. Now let's take a look at these three words that you may encounter on your reading: your hypertonic, hypotonic, and isotonic solution. So when you say hypertonic, hyper means high. There's a high concentration of solute relative to another. For example, the cell cytoplasm. When the cell is placed in a hypertonic solution, the water actually diffuses out of the cell, causes the cell to shrink or shrivel. Now, hypotonic means it has low concentration of solute relative to another solution. When a cell is placed in a hypotonic solution, the water actually diffuses into the cell, causing the cell to swell or possibly explode. And isotonic solution, iso means the same or equal of solute as another solution. And so when the cell is placed in an isotonic solution, the water diffuses into and out of the cell at the same rate. Therefore, the fluid that surrounds the body of the cell is isotonic. Now let's take a look at osmosis in terms of concentration. So let's say, for example, we have a hypertonic solution. So the water solution outside of the cell is saltier than the inside of the cell, and therefore there's more. Okay, this will cause the cell to shrivel and shrink. For example, pouring salt on a slug will actually cause it to shrink because the outside of the cell is saltier. So that's an example of hypertonic. Let's go to hypotonic. So when the water solution outside of the cell is less, the hypotonic solution will cause the cell to take in that water and swell. So that is hypotonic. So the solution is actually more on the outside, and therefore the tendency of the water is to go in to the cell. And lastly, which is your isotonic, the water outside the cell has an equal amount of salt as the water inside the cell. Okay, so it's equal. So it means there's no change in the cell that's happening in an isotonic solution. So again, hypotonic, less outside, more inside. Isotonic, equal. Hypertonic, more outside, less inside. So in terms of the red blood cell, as you can see here, if it's in hypertonic, it shrinks. Isotonic, equal. Hypotonic, it swells. So that's just a quick visual of your hypo, iso, and hypertonic and red blood cell. Now let's go to active transport. So the active transport uses energy in the form of ATP. It can be exocytosis, endocytosis, pinocytosis, or phagocytosis. So what's the difference? Exocytosis means how materials exit the cell, or how the cell uses the bathroom, as you call it. Endo means how materials enter the cell eating or engulfing, as you call it. The pinocytosis or pinocytosis, how small materials enter the cell, which also the same as eating or engulfing. And phagocytosis, how larger materials enter the cell by cell eating or engulfing. So in active transport, the movement of the molecules is from low to high concentration. Think of it this way: it takes more effort to really push something if there's too low of concentration going to the high concentration, right? So energy is required as molecules must be pumped against the concentration gradient. So proteins that work as pumps are called protein pumps. For example, the body cells must pump carbon dioxide into the surrounding blood vessels to be carried to the lungs for exhalation. Now, the blood vessels are high in carbon dioxide compared to the cell, so the energy is required to move the carbon dioxide across the cell membrane from low to high concentration. So that's an example of active transport. Now, just to summarize and kind of compare passive and active transport. Active transport is like going uphill, and your passive transport is like going downhill because there's no energy needed. And in passive transport, we have your diffusion, osmosis, and facilitated diffusion, whereas your active transport includes exocytosis, endocytosis, pinocytosis, and phagocytosis. I hope this helps a little bit for you to understand cell transport. Please read on, and I will see you in class. Bye.